All right, guys. So I am going to be doing a lot of tissue culture transfers today. We're going through our whole inventory. I'm getting ready to turn over all my cultures for 2021, and I wanted to try a couple different techniques this time. So Justin, who had sent these handles, um, he also sent me some cleaning solution that I'm going to be trying out experimentally. We got some chlorhexidine solution. It looks like it's veterinary cleaner, and I've got my map torch today, so I'll be sterilizing some blades using flame sterilization. And we've got this solution here, which is designed to help mitigate contamination. So this is a um, chlorhexidine gluconate, and it's a disinfectant. It has alcohol, blue one, citric acid, water. I assume that that's some kind of surfactant, but essentially it's used in lieu of the sterilization pouches, which I'm accustomed to these sterilization pouches. This is what I always use. So I'll kind of compare the difference between this cleaning solution for tissue culture and our sterile packs. So one of the main reasons why we go through all of our cultures every year is just to change over um, onto fresh agar so that the mycelium will still be healthy. So we've got a few different types of media. So this is V9 agar. I'll open it up for you so you can see. Um, but I like to use V9 for this purpose of bringing back the mycelium. So we've got some V9 here. You can see how orange it is in comparison to the MEA plates. There's a lot more nutrients in here, and it really helps the mycelium come back to life when you're transferring. So these are all of our different strains here. We've got all these slants of our cultures that we store long term, and we're going to be transferring these over to these new slants here, as well as the V9 agar, and we've got MEA or malt extract agar plates over here. So if anyone has any questions, feel free to chime in. I'll do my best to answer questions as we go. So I'm just going to start working. And the first thing, I'm just going through all my plates to make sure that there's no contamination. These are about a month old, so I just want to double check. So they look clean so far. So if you're just joining in now, we're just doing our plate transfers, and this is V9 agar, which is a V8 juice and potato flakes, and it's used to revitalize the mycelium after a long year of being replicated over and over again. So it's always good to switch your mycelium up on fresh media, and we like to use V9 agar, or you could also use potato dextrose, um, trypticase soy agar is a good one too, or MEA like we have over here. Alright, so I've got some V9, MEA, and these are our cultures. So I'll go through. This is our brown oyster. It's one of my favorite oyster mushrooms. It does really good in, you know, pretty much every temperature. You can see how healthy that one is. It's got really nice growth and it pins in like 4 days once you transfer into fruiting. So this is one of my favorite strains. You can find it on our Etsy, Fresh Fungi. So we are going to open up a fresh blade here, and I'll start by using my sterile handles that I normally use, and I'll do a few transfers with my normal technique, and then I can switch over to this blue cleaning solution that Justin Langley had sent me in the mail. So shout out to Justin. Sorry, I was busy. I wanted to do this video a little sooner, but here we are. So I'm just peeling off the parafilm right now. So MEA is a really highly nutritious media. It it is pretty prone to contamination. So, I usually pour about 500 a week and get 1 or 2 or 3 contaminated with yeast usually. So it's pretty common to get MEA contaminated. You can also add antibiotics, which might help, but I don't like to use antibiotics. I feel like it's just a cover up, but it does help if you don't have a flow hood or if you're having some problems. Potato dextrose is another good one. Alright, so I've got my brown oyster right here, and I'll transfer one onto the V9, and then I like to have a few backups for in February when we start to get things going again. So and you can see how small wedges I'm cutting here. So technically I could probably get about 40 or 50 isolates or expansions from one single plate. But I don't need to do that today. Alright, so I just pulled this slant here and notice there's some rogue mycelium here. So that's why you always want to leave your plates and your slants out for a few days. I don't know what mycelium that is, but that's not sterile, so I'm not going to use this one. But you can see how clean that one is. So just always be aware of what you're doing. Alright. And now I like to hold the cap kind of loose in my hand. And then I'm just going to take my wedge here. Alright, and then I'll label these so that I don't get too far ahead of myself. It's always good to just, slow down and label. And normally I'll label on the underneath, but because I just transferred that agar, I don't want it to flip over. So I'll just label these on the lid. So for everyone that's just joining in, we are just doing our transfers to create our fresh culture library for 2021. So I just pulled from our brown oyster and I used um, the sterilized blade from the pouches, and now I'm going to switch over and try a different technique. I'll just set this. So a quick rack that you can use if you don't have a test tube rack, I usually will just grab a jar and you can set your slants up like that. Alright, so this is the first time I'm going to be using this technique. So these are the blade handles by Justin Langley. If you watched our video from a couple weeks ago, he makes these custom stainless steel handles, and he had reached out to me about a different procedure that they use over at their farm. So I'm going to test it out and just see how I like it. Alright, so I've got our brown oyster here, and this has been sterilized in this blue solution. And now I'm just going to cut a wedge out of here. I prefer to use the number 11 blades, but that worked pretty fine. So the pro about using this technique with the sterilizer solution and the blowtorch is that alright, so someone asked, is that alcohol? So this is chlorhexidine. It's alcohol with a surfactant in it. So it's used in tissue culture, and it disinfects the blade handle. And then we're going to use this map torch to sterilize this blade. So this culture is fairly new. It's probably about a week old. I just pulled it from our slant, these are all of our G1 cultures. So before I transfer my slant to store for long term, I grow it out on agar so that I know that it's clean and healthy. So these slants are about 8 months old. This culture is about a week old. I keep these refrigerated all year, and then I pull them out to expand my cultures. So then it grew out on this plate, and now I inoculated a slant and a few more petri dishes that I'm going to be storing over the winter. And we're also going to have these available for sale on our Etsy. So if you haven't checked out our Etsy, it's fresh fungi, and there's a bunch of liquid cultures. But alright, so I'm going to be switching over from these brown oysters, and now we're going to go to a Boletus edgeless. So this one's from 11.6. I've got some really exciting plans for Boletus edgeless or porcini, which this is a mycorrhizal mushroom, but there's a lot of studies out that have shown it to be compatible with a rock rose species of It's a bush with a flower, and then in combination with some bacteria, Pseudomonas fluorescens is one of them. So I'm planning on doing an outdoor garden bed with these in the upcoming spring. So here's our Boletus edulis, and I'm going to use the flame sterilizer to transfer this onto some V9 agar, as well as a slant and some MEA over here. So this tool has been sitting in the sterilizer solution for about 20 minutes. I took it out and put a fresh number 10 blade on there. So now I'm going to carefully turn on my map torch here. Alright, so I'm just going to be heating up that blade till it gets red hot. So I transferred this onto a liquid culture, so I'm just putting this blade down to cool it off before I make my transfer. But so I did make some liquid cultures. They're available on our Etsy, and you can transfer it onto grain. But the one problem with Boletus edulis is that it's a mycorrhizal species, so it requires a relationship with the root of a plant to provide the nutrients necessary for it to fruit. So there's some really exciting papers that have come out of France, and it seems to be possible to get some fruits with a couple rockwool plant species, or you can inoculate in the roots of the tree. This bolete was found by a hemlock tree, so conifer species, but you can also grow them with oak trees. And let's see the results. So I'm gonna take this blade off and switch over to my sterilized blade from the pouch, and I'm just gonna flame sterilize this so we have a control. So I really like the length of that other blade. I'm not getting closer or very close to the flame when I use the other blade. And I'll just do a comparison here. But one thing when you're using flame to sterilize, you always want to cool your blade off so that you're not killing the mycelium. When you're making your transfers. Alright, so I've got my Boletus. I'm going to turn off this flame here. While I label these. So I'm just going to label I'm going to mark it as a G2 because it's the second generation from the original clone. And then 12, 4, 20. And then this one's going to be the short handle. So I'm just comparing if there's a difference between sterility using both of these techniques. But I feel like there's a lot of pros and cons to using this alcohol solution compared to the sterilization pouches. It's just personal preference. or, you know, what you have available in your lab. So I'm going to transfer one more onto a slant. So I'm just checking all my agar, make sure my cultures are clean. And whoops. All right. So if that happens, it's always good to have some alcohol, and you can start over. All right. I've also got some cordyceps that I'm going to be pulling some fruiting bodies onto V9 to preserve that phenotype. I'm going to be running some more cordyceps over the winter. So someone asked, is the liquid culture just a normal honey recipe? Yes. I have also been messing with some TSB, which is tryptase soy broth. It's just sugar and soy in a solution, and it's just a good way to mix up the substrate to keep your mycelium healthy. But you can buy lots of different medias. I like buffered peptone sometimes. That's a good liquid media. And then this is on V9 agar, which is V8 and potatoes. All right, so that is the experiment comparing these long handles to the short handles and sterilization pouches compared to the chlorhexidine solution. So I'll keep you guys posted on the updates. I've got a lot of cultures to go through here. I'll just continue working for a little bit. If anyone has any questions, feel free to drop a comment. I've got it set up so I can read those. I've got a new bear's head. So this is Hericium americanum. So that's what's up next. I'm going to be transferring this guy onto V9, a slant, and some MEA. So I'll just stick this in the blue solution here, let that soak. Got another one of these, and I'm just going to use my pouches until these are out, just because I'm more comfortable. I also actually, so I've got some black morel, so Morchella esculata. I've got some spores in here that I made from some dried morels. So I'm going to actually streak this out. I'll show you guys how I do that before I go on to the bear's head. So this is actually pretty exciting. I want to get some more morel patches going in my yard. And the first thing you have to do is get some good isolates. So I'll get this flame going again. Howdy. So I am in the middle of transferring all my cultures to restock my inventory. These are going to be available on Etsy on Fresh Fungi. And right now I'm about to streak some black morel spores. So I got these from some dried mushrooms. It's some morchella mushroom. And right now I'm just going to heat up my loop. Alright, so I'm just letting the loop cool off. You can also put on some agar so it cools. And I'm going to take my loop and do a 3-part streak here. So you can see the water droplet in the loop, and that is going to be carrying the mushroom spores. And now what you want to do is, I shouldn't have shut the flame off, but right now there's a high concentration of spores in that area that I streak. So right now I'm going to be eliminating all the spores that are left on the loop. And then I'm going to go back to this plate and then grab the spores from that section. And then I'll repeat that one more time. And that's called a 3-part streak. And the idea is that you're going to end up isolating some spores in that last streak right there. So I'll do that a couple times. Up. So that was a little too much. But that's okay, I'll make it work. Alright, maybe we'll just do 2 plates for now. But alright guys, so I'm going to continue doing this work on my own, but stay tuned for more Fungi Friday. We're going to be doing these regularly on Fridays now, and I've got some guests that I'm going to be bringing on, and I want to do like some kind of podcast style mycology video, but also do lab techniques mixed in. So if you have any suggestions, leave a comment. And if you're looking for some high quality genetics, check out our Etsy at Fresh Fungi. Alright guys, until next time, much love.